Good afternoon one and all. So here we are in our third session from Microbial World, wherein in our previous class we have dealt with the multiplication of bacteria. We have gone through clearly through lytic cycle and lysogenic cycle and types of bacteria, and then we have also looked into viral diseases, bacterial diseases completely, plant viruses and animal viruses, bacteriophages, totally we have looked into. So in today's session we are going to start the class with bacterial cell structure, learning its structure, physiology and nutrient diversity. Okay. So which kingdom is highly metabolically diverse is Monera. Monera is the kingdom which is metabolically diverse, where it has many types of nutrition and respiratory pathways. Okay. So in nutrition, if you have to consider, they can be autotrophic or heterotrophic. Autotrophic, again they can be photoautotrophic or chemoautotrophic. If you have to look into the respiration of bacteria, they can be obligate aerobes, facultative anaerobes, obligate anaerobes or facultative aerobes. Reproduction, sexual recombination is of three types in bacteria, which includes conjugation, transduction and transformation. So we will be looking into all these concepts today. Okay. Let us get started with bacterial cell structure. Bacteria does not just have a cell wall, but bacteria is surrounded by cell envelope. Okay. So bacteria has outermost cell envelope. This cell envelope will have what? Cell envelope will be having, the outermost layer is glycocalyx. Okay, this is the first outermost layer of the cell envelope in the bacteria, that is glycocalyx. Glycocalyx can be loosely arranged or tightly compactly arranged. If it is loosely arranged, it is called as capsule. Sorry, it is called as slime layer. If it is tightly arranged, it is called as capsule. Fine. So yes, the outermost layer of the cell envelope is glycocalyx. If it is loosely arranged, you will call it as slime layer. If it is tightly arranged, you will call it as capsule. Okay. So the outermost layer is about glycocalyx, that is clear to us. Next to the glycocalyx, the second layer in the cell envelope is cell wall. Cell wall is made up of, in most of the bacteria, cell wall is made up of what? Peptidoglycan. Cell wall is made up of peptidoglycan, and the innermost layer of the cell envelope is cell membrane or plasma membrane. I hope the cell envelope is clear to everyone. The outermost layer is glycocalyx. If it is loosely arranged, you will call it as slime layer. What is the benefit that bacteria is getting if the outermost layer is slime layer? It will prevent the bacteria from loss of minerals and nutrients when it is exposed to the extreme habitats like halophilic conditions, thermoacidophilic conditions. In such conditions, the bacteria is prevented from the loss of water and minerals if the outermost layer is slime layer. So if it is slime layer, it will prevent the loss of water and minerals. If it is capsule, okay, the glycocalyx is thickly arranged, the outermost layer is capsule. What is the benefit that bacteria is getting if the outermost layer is capsule? It will hide from the host immune system. Bacteria, whenever it is infecting any host, from the host immune system, this is a protective sheet where this hides the bacteria from host immune system, and this will also give resistance to antibiotics. These two are the major benefits if the outer layer is capsule. What are they, ma? First is resistance to antibiotics. Second will be hides from host immune system. Host immune system. Clear, right? So, this is about the cell envelope in bacteria. Now, the innermost layer, that is plasma membrane, this has extensions. This has extensions inner to the cytoplasm. Those extensions are called as mesosomes. Very important structure, mesosomes, quite a times asked in the previous year exams. Okay. Mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. What are the major benefits of the mesosomes? What are the major functions of the mesosomes? Mesosomes special function is respiration. This invaginations or inclusions of cell membrane towards the cytoplasm can be in the form of vesicles, it can be in the form of tubules, it can be in the form of lamellae. So, the extensions of cell membrane or inclusions can be in the form of vesicles, tubules or lamellae. Fine. These invaginations primary function is to release the respiratory enzymes, to promote secretion, to help in cell wall formation, daughter cell replication and cell division. Okay. So, all these are the functions of mesosomes. One of the major function is respiration. That is the reason it is said as mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Now, when we have to consider this fluid filled matrix inside is what? Cytoplasm. Cytoplasm constitutes what? As it is a prokaryotic cell, there will not be any membrane bound organelles, but then non-membrane bound organelles are present. What are the non-membrane bound organelles which are seen in the cytoplasm? Ribosomes. Ribosomes, which are 70S type of ribosomes. 70S type of ribosomes, wherein larger subunit will be 50S, smaller subunit will be 30S. Where are these ribosomes actually present? Ribosomes are present inside the cytoplasm and they are present attached to the cell membrane. So, these are the two locations of the ribosomes. The primary function of the ribosomes is protein synthesis. Okay. Primary function of the ribosome is what? Protein synthesis. So, those ribosomes which are present in the cytoplasm are involved in protein synthesis. Those ribosomes which are attached to the cell membrane are involved in the protein synthesis which are for translocation. Clear, right? So, this is about ribosomes. Apart from ribosomes, inclusion bodies are present in the cytoplasm. Okay, what are they? Inclusion bodies. These inclusion bodies are nothing but the storage materials inside the prokaryotic cell. Inclusion bodies are also non-membrane bound organelles. Some of the examples of inclusion bodies include cyanophycean granules, phosphate granules, sulfate granules. These are the examples of inclusion bodies. Now, it is clear to us, right? The outermost cell envelope is 3-layered. Outer layer is glycocalyx. If it is loosely arranged, it is called as slime layer. Tightly arranged, it is called as capsule. If it is slime layer, this will prevent the bacteria from loss of water and minerals. If the outer layer is capsule, it will prevent the bacteria to hide from the host. It will help the bacteria to hide from the host immune system, or it will provide the resistance to antibiotics, right? The second layer of the cell envelope is cell wall, which is made up of peptidoglycan, and the third is plasma membrane or cell membrane. Plasma membrane or cell membrane will have inclusions or invaginations towards the cytoplasm. They can be in the form of vesicles, tubules or lamellae. These inclusions are called as mesosomes. There is a most important question, what is that? Mesosomes of a prokaryotic cell are equivalent to which organelle in the eukaryotic cell? It is equivalent to mitochondria of a eukaryotic cell, and this majorly promotes what? Respiration. What are the other functions of the mesosomes? Respiration, secretion, cell wall formation, DNA replication and daughter cell formation. These are the 5 major functions of mesosomes. Then when we look into the fluid filled matrix, that is cytoplasm, inside cytoplasm we have 70S ribosomes which are non-membrane bound, whose larger subunit is 50S and smaller subunit is 30S. There are 2 locations or sites of the ribosomes. One is inside the cytoplasm and second is which is attached to the cell membrane. 70S ribosomes attached to the cell membrane. This ribosomes which are in the cytoplasm will help in protein synthesis. Those ribosomes attached to the cell membrane will help in translocation of the proteins whichever are synthesized. Clear, right? Another non-membrane bound organelles which are present in the cytoplasm include inclusion bodies. Okay, these are what? Inclusion bodies. Inclusion bodies will store the reserve food material. So, they can be cyanophycean granules, phosphate granules or sulfate granules. This is about the non-membrane bound organelles which is present in the fluid filled matrix. Now, when it comes to the genetic material, okay, so I hope this complete thing is clear to you. I will speak about the genetic material now. The genetic material which is present in the bacteria, just a second, yes. So, the genetic material which is present inside the bacteria, this is of 2 types now. One is called as genomic DNA or chromosomal DNA, this complete thing which is called as what? Genomic DNA or chromosomal DNA. Take down genomic DNA or chromosomal DNA, which is double stranded circular DNA, fine, whose main target is to focus on the complete metabolic activities that are taking place in the cell. So, this majorly focuses on the metabolic activities which occur inside the cell. Okay. The second DNA is called as extra chromosomal DNA or plasmid DNA. Okay. Second DNA which is present inside the bacterial cell in the cytoplasm is the plasmid DNA. Okay. Plasmid DNA or extra chromosomal DNA. Extra chromosomal DNA. So, this plasmid DNA or extra chromosomal DNA is again double stranded circular DNA. Okay. Plasmid DNA, extra chromosomal DNA is again what? Double stranded circular DNA. This focuses mainly on few functions like giving a particular phenotypic character to the bacteria, providing resistance to antibiotics, and most importantly conjugation, conducting the sexual recombination, one type, that is conjugation. These 3 functions are modulated by plasmid DNA which is present in the bacteria. So, the first is what? Plasmid DNA or extra chromosomal DNA. It will provide a particular phenotypic character to the bacteria, phenotypic character to the bacteria, and the second is providing resistance to antibiotics, and third major important function of plasmid DNA is to promote conjugation. Fine. This is the complete structural description of a prokaryotic cell from the cell envelope to the genetic material. Okay. So, each and every point which I have discussed should be highlighted and should be revised once again, because many times the questions appear from that. Okay. So, the clear bacterial structure is evident to us after the discovery of electron microscope, which revealed the bacterial structure is more complex than small spheres or rods. When we have learned types of bacteria based on their shape, we have learned the spherical bacteria, rod shaped bacteria, bacillus, comma shaped vibrio. More than that, shapes, bacteria has this complex structure inside is being understood after the discovery of the electron microscope. Now, So here if you see flagella, we have many types of flagella. Based on the flagella, we have different terminology. So if the bacteria has only one flagella, if it has only one flagella at one end, this is called as monotrichous, one polar flagellum. Second is amphitrichous. If one flagellum at each end, one flagellum at each end, that is called as amphitrichous. If bunch of flagella is present at one end, then it is called as lophotrichous. Okay, bunch of flagella at one end, that is lophotrichous. Flagella is distributed all across the body, then it is called as peritrichous. Okay, so one polar flagellum, that is monotrichous, one flagellum at one end, one flagellum at the one end, that is amphitrichous, bunch of flagella at one end, that is lophotrichous, peritrichous where flagella is completely distributed all across the body. So now I would like to discuss about the motile and non-motile structures of the bacteria, which are again important. Okay, we have motile and non-motile structures of bacteria. Motile and non-motile structures. So the only motile structure which is present in the prokaryotic cell is flagella. The only motile structure is flagella. This flagella will have what microtubular arrangement? 9 plus 0 microtubular arrangement is present in the flagella, which is the motile structure. So this flagella has three parts. One is what? Basal body, basal body. This is filament, filament and hook. Sorry, basal body, hook and filament, hook and filament, which has 9 plus 0 arrangement. So the only motile structures which help in the movement or locomotion of the bacteria is flagella, that is 9 plus 0. Cilia are not present in the prokaryotic cells. Cilia are present in the protozoans, which are unicellular eukaryotes. Next is non-motile structures. Non-motile structures include pili and fimbriae. Okay, pili and fimbriae, these are the non-motile structures. Okay, pili will help in the, pili are nothing but the straight pointed structures which will help in the attachment to the substratum, and pili will also act as sex pili to conduct the sexual recombination, that is conjugation, which will link up between two different strains of bacteria, that is F plus and F minus strain. So to promote this conjugation, okay, to promote the conjugation, sex pili is required. So this is tubular or elongated tubular structure you can describe as. Fimbriae are nothing but bristle like structures sprouting out of the cell wall. Okay, fimbriae are bristle like structures sprouting out of the cell wall. They will help in attachment to the substratum. They will help in attachment to substratum. Fine. So with this, the complete structural description of bacteria is done, including motile and non-motile structures. Next. And flagella is also discussed. Now let us look into the plasmids. Just a second. Yeah. So apart from the genophore, as we have discussed, what is genophore? Bacterial chromosome, which is also called as genomic DNA or chromosomal DNA. Apart from this, yes, Shivakumar sir, it is, the genomic DNA is circular. Genomic DNA is double stranded circular DNA. Okay, plasmid DNA is also double stranded circular DNA. Fine. So plasmids, apart from the genophore, main bacterium chromosome, bacteria is also contains plasmids, which is circular, double stranded, which is also described as extra chromosomal DNA. So the synonyms are important: genophore, bacterial chromosome, genomic DNA, chromosomal DNA, one and the same. Plasmid DNA, extra chromosomal DNA, non-genomic DNA is one and the same. For your better understanding, I will write at a place. Yes. So genophore, bacterial chromosome, genomic DNA or chromosomal DNA is one and the same, and here extra chromosomal DNA, plasmid DNA, non-genomic DNA, is one and the same. Okay, take down. Next, characteristics. Fewer genes than the genophore. Characteristics, major characteristics is they have fewer genes than the genophore in plasmid DNA. They often provide resistance to drugs, toxins, enzymes and antibiotics, and then they can be gained or lost without harming the bacterial cell. So, plasmid DNA can be gained or lost without harming the bacterial cell. This is some description about the plasmid DNA in specific. Now, let us look into the metabolic diversity. Okay, I already told you which kingdom in the 5 kingdom system of classification will exhibit maximum metabolic diversity. So, the maximum metabolic diversity is exhibited by the kingdom Monera, that is prokaryotic cells. So, this metabolic diversity includes what? What is metabolism? It includes both anabolism and catabolism. Anabolism and catabolism. Anabolism includes nutrition and catabolism includes respiration. Anabolism includes nutrition, catabolism includes respiration. Nutritions, if you have to take, we have autotrophic nutrition, heterotrophic nutrition. Autotrophic, again we have photoautotrophic nutrition, chemoautotrophic nutrition. Heterotrophic, we have 3 types. What are they? Symbiotic type, saprophytic type, and next is parasitic type. So, the bacteria will exhibit all these modes of nutrition. That is why it is exhibiting high nutritional diversity. In respiration, if you have to see, we have aerobic respiration, aerobic and anaerobic respiration. Aerobic respiration, we have obligate aerobes or facultative anaerobes. To remember facultative anaerobes and facultative aerobes, there is a small trick. Facultative means nothing but sometimes. Okay, sometimes it is acting like anaerobes, means most of the time the food is oxidized only in the presence of oxygen, but whenever oxygen is not present, whenever the bacteria is deprived of oxygen, at that time also it is oxidizing. So, that is why it is called as facultative anaerobes. Sometimes it is acting as anaerobes. And then anaerobic respiration, we have obligate anaerobes or facultative aerobes. Obligate anaerobes or facultative aerobes. Till here it is clear, right? Next, bacterial nutrition is determined by source of carbon and source of energy. Photoautotrophs. What is meant by photoautotrophic nutrition? Photo means light. So, using sunlight or light energy, the food is prepared, is called as photoautotrophic nutrition. Here, carbon dioxide is fixed into glucose. Carbon dioxide is fixed into glucose. Best examples include chromatium and chlorobium. Chromatium, chlorobium, these are the best examples. And then if you see chemoautotrophic nutrition, chemo means chemicals, okay, chemicals or inorganic chemical constituents. So, here the energy is the light energy, but chemoautotrophic nutrition energy is from ATP. The energy source to prepare the food material is ATP, and the inorganic elements like sulfur, nitrogen, hydrogen, ferrous, these are the electron donors over here, which will fix the carbon dioxide. Best examples include Nitrosomonas, Nitrobacter, Bigiotova and Methanogens. So, here what will be the electron donors here? Nitrosomonas, Nitrobacter will be nitrates and nitrites will be the electron donors. Bigiotova sulfates will be the electron donors, methanogens, methane will be the electron donor. In photoautotrophic nutrition, water will be the electron donor. One added advantage of chemoautotrophic nutrition is it plays major role in the mineral cycling. Okay, chemoautotrophic nutrition, it plays major role in mineral cycling. Mineral cycling. Clear, right? So this is all about the metabolic diversity. First we have exercise, that is nutrition in bacteria. Of them, photoautotrophic and chemoautotrophic is clear to us, right? Next. So here if you see photoheterotrophs and chemoheterotrophs. So till today's session we will finish up to metabolic diversity, and tomorrow we will be looking into the sexual recombination. Okay. So photoheterotrophs, they will use light for energy, but carbon is derived from organic compounds. Unlike chemoautotrophs, here in photoheterotrophs, carbon is from the organic compounds, their carbon is from the inorganic elements. For example, rhodospirillum, rhodopseudomonas, these are the best examples for photoheterotrophs. In all the scenarios, the mode of nutrition and its example is very much important. And then chemoheterotrophs, they use organic molecules for both carbon and nitrogen. Organic molecules are used both for carbon and energy. Next, energy from respiration or fermentation, that is ATP production. Energy is taken from what? Respiration or fermentation, that is ATP. So in chemoheterotrophs, strictly ATP energy is only used, and carbon is from the source of organic molecules. Both in photoheterotrophs and chemoheterotrophs, carbon source is the organic compounds. And next, chemoheterotrophs. Chemoheterotrophs, we have saprophytes and parasites. So here if you see, saprophytic nutrition is they will feed on dead and decaying matter. How does they feed on detritus or dead or decaying matter? See, if at all this is the bacteria, this is the bacteria, and here is there is some complex food material, here there is some complex food material. This bacteria will be releasing out the enzymes, okay, hydrolytic enzymes. This hydrolytic enzymes will digest the complex molecule and it will turn to simpler molecules. These simpler molecules will be taken by the bacteria. Clear, right? So here bacteria are releasing out the enzymes, complex molecule will be break to simpler molecules, simpler molecules will be absorbed by the bacteria. So here saprophytes will feed on dead organic matters, that is detritus, best example is bacillus. Parasites, they derive nutrients from host living cells and tissues. Okay. Example include Xanthomonas and Salmonella. Next. Yes. So with this the nutrition part is done. Reproduction we will be looking in the next class. Reproduction, sexual recombination, which includes conjugation, transformation, transduction. We will deal with experiments in detail in the next class. So that's all for today's session, and thank you.